SOS repair system is controlled by the SOS operon, which is activated upon DNA damage. Under normal circumstances, when the DNA is not damaged, the REC-A protein is inactive. REC-A protein is an inhibitor of LEX-A protein. Thus, since REC-A is absent, LEX-A protein accumulates. LEX-A protein binds to the promoter region of the SOS operon. This leads to the repression of all SOS responding genes. Thus, when the DNA is not damaged, the SOS response system is off. When the DNA is damaged, the REC-A protein gets activated, and this activated REC-A protein inhibits the LEX-A protein by cleaving it. The cleaved LEX-A protein cannot bind to the promoter site. Thus, there is no repression of the SOS operon and the SOS response system is induced. The SOS genes are transcribed and translated to repair the damaged DNA.
If this video was helpful, then please like this video, share it with your friends, subscribe to my channel, and don't forget to leave us your valuable comments. Thank you.